Good evening to you. Hello. Okay. Hello, everybody. Welcome back. And this is the week 8 question answer session of the NPTEL course Fundamentals of Protein Chemistry. This course is coordinated by Professor Dasgupta from the Department of Chemistry, IIT Kharagpur. I am Vyas Mukherjee, a research scholar from the Department of Bioscience and Biotechnology, IIT Kharagpur. And in this class, we'll be talking about metalloproteins and motor proteins. Okay. Now, metalloproteins and motor proteins are two entire huge areas of research, and so whatever I can tell you based on the question and answers that we have to discuss today, I will give you the information. And in the slide, there is a lot of additional information for you to go through. Okay. Besides this, the books have been provided, research articles, review papers, and a lot of website links, YouTube videos, everything has been provided. Please go through all of these, and then if you find these topics very interesting, you can go deeper into these. Okay, so today we will start with the question answer session. All right, so we will be talking about metalloproteins and motor proteins. Now, metalloproteins are proteins which have metal ions or metal atoms as a very fundamental element in their structure. And motor proteins are the proteins that help in mobility. Okay, so we'll be talking about these two, and there are many examples of metalloproteins and motor proteins in our body, in plants, in bacteria. All right, and we'll have a look at all of those. Okay, you can ask me questions, and you can also write down in the chat box and we can discuss. Okay, if you're having any doubts with anything, if you have anything else to convey, we can discuss that way. Okay, so let's start with this metalloproteins and motor proteins. So metalloproteins, as you know, these are metal containing proteins, and the metal atom or metal ion can be zinc, iron, copper, manganese, cobalt, etc. This is very, very essential for the protein's activity, stability, and overall function. Okay, this is an integral part of the structure of the protein sometimes. All right. So, for example, we have hemoglobin, ferritin, cytochrome C, carbonic anhydrase, etc. We will look at all of these in details very soon. Okay, and now what about the motor proteins? So if you look at the figure on the left, you can see that there is a protein. This is having a tiny metal ion. So that green one that you're seeing, that is the metal ion, and there are residues that are actually binding to the metal ion. It is keeping it in place, and this metal ion is actually helping in the activity and the structural conformation of the protein. Okay. The figure on the right, rather, this is actually showing motor proteins. So motor proteins are a very interesting class, and if you actually see the video that I have attached at the end of this PPT, you will see that this motor protein actually walks. Okay, this actually walks on the microtubule of your cell, and it carries different kinds of cargo. Cargo means goods, right? So different kind of cargo it is carrying. So here if you see, this is kind of a vesicle. Now inside that vesicle there can be proteins, there can be anything, okay, any lipid, carbohydrate, and then this is actually carrying that vesicle and it is walking. So if you see those tiny orange feet-like structures, those are actually the structures used for walking. Okay, this is called treadmill. So we will be looking into that soon. Now, motor proteins are proteins that convert chemical energy by ATP hydrolysis into mechanical work. That is how they work for cellular processes, including movement, transport of cellular materials, and very importantly, cell division. Okay, right. So now we will move on to the different functions of metalloproteins. Now, metalloproteins are involved in wide range of functions that includes photosynthesis, respiration, chemical reactions, redox reactions, signal transduction, DNA synthesis, and the transport of different materials across the cell. It is also involved in cell division. Okay. Okay, so this is all about metalloproteins. Now here are some examples for you to go through. There is hemoglobin, which you know contains iron. There is myoglobin, of course it contains iron. There is ferritin and cytochrome C. All of these are containing iron in them, right? And then there is other protein. There are other proteins like superoxide dismutase, which has so many different ions in them, right? So similarly, you can go through all of these and you can, if you're interested, you can remember what metal ion is contained in them, okay, for your curiosity. Now, one very interesting question is, while reading about enzymes, I'm sure you guys remember that we had talked about the enzymes with cofactor. Right, cofactor is also often metal ion. Now, what is the difference between this metalloenzymes and enzymes with metal ions as cofactor? Well, the enzymes that are activated by metal ions or cofactors, these actually come under a group of these metalloenzymes. So most often metalloenzymes contain this iron as an integral part of their structure. Okay, but then for the enzymes, the bond is relatively weaker with the cofactor and it is only required for its enzyme activity. It is not required for its overall structure and stability as such. Okay, so that is one of the differences. You can look up and you can find more differences, okay, if you're interested. All right. So now this is something very interesting, and we say that it's a blood red rose, right? We always associate the color red with blood. However, if you look in nature, there are different colors of blood found in different organisms. So very interestingly, if you're a non-vegetarian and if you like to have prawns, you will know that prawns are said to have blue colored blood. Okay, they do not have this normal red blood that we do. And the reason for that is our blood is having hemo, what, globin, that is giving it the red color. But then the blood that you see in crustaceans and mollusks and octopuses, so they actually have hemocyanin, which is a different compound, and this is made up of copper instead of iron. Okay, and this gives it the characteristic blue color. Next, certain worms even have green color and violet color blood because of the presence of chlorocruorin and hemoerythrin. These are different compounds containing iron. Okay, but then the porphyrin ring is not there in case of hemoerythrin, and this is how different pigments give different color to blood in different organisms. Okay, so we will look at this hemocyanin in details. Okay, so before that, we will have a look at the different metalloenzymes found in our body. For example, hemoglobin, of course you remember hemoglobin, this is having this porphyrin ring structure and iron is there present, and this is the oxidation state of iron, which is +2, right? So, I'm sure you remember myoglobin as well. It is having again porphyrin ring structure with iron. Okay, and then there is horseradish peroxidase. Horseradish peroxidase is a very common enzyme used during Western blot, right? So this enzyme also has porphyrin ring with iron, and it is also having a histidine molecule bonded to it. And then there is catalase enzyme. So you can look at catalase structure. There are manganese in its structure. Okay. Now we will have a look at hemocyanin. So hemocyanin looks like this. There are two copper ions bound to it, and deoxyhemocyanin contains Cu1 plus ions, 2 Cu1 plus ions per subunit, and it is colorless. Whereas when it binds to oxygen, it becomes blue in color. Right. So this is very interesting thing that you must remember. And also its oxidation state changes from +1 to +2. Okay. So that is something that you also need to remember. And this is majorly seen in you have seen already, octopus, snails, squids, etc., and also arthropods. Okay, now we will look at some other metalloproteins. The majority of these are found in plants. Okay, so iron containing proteins are there, which are mostly heme proteins. There are also others, and there comes in the iron sulfur proteins. So iron sulfur proteins, if you remember, they are very active in our mitochondria for the electron transport chain, as a part of electron transport chain. So that is useful during what? Tell, if you remember, what is electron transport chain and where does it come handy? We have studied about electron transport chain earlier during our school in two different places. One is photosynthesis and the other one is respiration. So in both of these cases, electron transport chain, yes, it generates ATP. That is right. Okay. So here it is very, very active. And these iron sulfur proteins actually come in handy during this electron transport chain. Plus, it is also useful in nitrogenase enzyme. So what is nitrogenase enzyme? If you know about these grams, okay, gram is like chole. So this is a leguminous plant. It's basically the roots of this leguminous plant contain kind of a bulb structure, and inside that bulb structure there is a lot of bacterial activity going on. So there are bacterial species like Azotobacter which reside in these root nodules of the leguminous plant, and it is involved in nitrogen fixation from the atmosphere directly into the plant. Okay, and that gives rise to protein in your pulses. So, for this nitrogen fixation, this nitrogenase enzyme is actually active. Nitrogen fixation and nitrogen release into the atmosphere. So, there again you have iron, sulfur, molybdenum proteins and iron sulfur proteins. Okay, we'll have a look at those soon. So there are different classes. There is replication, peridoxin. Peridoxin is found in plants. Peridoxin is found in bacteria as well. Okay. So now there are cupredoxin proteins. So cupredoxin is a small family of copper-containing proteins that play a very important role in the electron transport chain. So if you look at all these four figures, I hope you remember plastocyanin. Plastocyanin is something that we encountered during photosynthesis, right? And also you might not be aware of this. This is azurin. Azurin is also a compound which is found in bacteria, that is Pseudomonas aeruginosa. Okay, and this is how azurin looks like. If you see the geometry of azurin, it is actually trigonal bipyramidal. So we will learn about azurin soon. All right, this is also involved in the electron transport chain. Okay. Now, to give you recollection, this is the Z scheme of photosynthesis. See, I was talking about these. Okay, one second. Someone has written something. It generates ATP, right? Okay. Okay, so if you look at this Z scheme of photosynthesis, you will see that there is plastocyanin. Plastocyanin is a metalloprotein, and we will learn about plastocyanin here. Plus, there are iron sulfur proteins, which again contain iron and sulfur, so definitely we'll be talking about them. There is also peridoxin, which we would be interested in. Okay, so these are the different proteins that you will read about today in today's class. Now we come to iron sulfur proteins. We just talked about this rubridoxin. So let's have a look at this properly. So see, iron with its iron plus 3 is a little distorted. So this is the structure of rubridoxin, and this is slightly distorted, and that is because of its environment. You can read about this in details if you are interested, but for today's MCQs, I think this much is sufficient. So reduction is not accompanied by a significant change in geometry and electron transfer component. This is actually found in purple sulfur, it is found in sulfur bacteria You can read about it, whether it is found in green sulfur bacteria or purple sulfur bacteria. There are two kinds of sulfur bacteria. So sulfur bacteria actually use sulfur instead of oxygen for their activity, right? So you can read about them. And so electron transport component of the sulfur bacteria. Now there is plant type pyridoxine. So in resting stage, 2 iron 3 plus, but only one of them is reduced to Fe2 plus. And this Fe2 plus, Fe3 plus, one unpaired electron is there and significant iron-iron interaction, but then complete electron delocalization is not happening. So this is the structure of plant pyridoxine. You can actually read about the structure of bacterial pyridoxine if you're interested. Okay. All right. Oh, acha. So this purple sulfur and green sulfur bacteria, these are actually chemoautotrophs. So that means they are using like they are very primitive life forms, and you can read about them. Okay. Right. So we come to the questions. So based on whatever we have learned until now, you must be able to answer the questions. Okay. You try to answer them in the chat box. Okay. So which of the following is false regarding hemocyanin? Okay. Right. We have very few students today. Right. So in the last class, there was a power shutdown in my area, and so we couldn't go about with the usual timings of the class. Screen is diminished. Oh, is that so? One second. Okay. I'll share it again. Let's see. Tell me if it is okay now. Can you see the screen? Is it okay now? You can answer in the chat box, and if it is not okay, you tell me, okay? Because it's very important that you understand whatever I'm saying, and for that you need to be able to see the slides properly. Can you see the question on the screen? Okay, it's visible. Great. So try to answer this question. So the first question is, which of the following is false regarding hemocyanin? It has two Cu ions in its active state. Cu ions are in the +1 oxidation state in the deoxy form. Each Cu is coordinated by two nitrogen from histidine residues. The deoxy form of hemocyanin is colorless. Now all the answers, all the statements are there in the PPT. Okay. So even if you're not able to remember now, you can always go back and you can check. Okay. So you're saying that C is the wrong answer. Let's see. Okay. Yes, that is very correct. If you go back to the structure of hemocyanin, you will see that every Cu atom is bound by 3 histidine residues, plus the oxidation state is +1 in deoxyhemocyanin and +2 in oxyhemocyanin. Also, deoxyhemocyanin is colorless, but when water is getting added, it is becoming blue in color. So very good. You have understood this, that means. Okay. Now we'll move on to some common metalloproteins in plants. Now we already read about plastocyanin, and we looked at the structures of what else? This one, iron sulfur protein, rubridoxin and pyridoxine. Okay. Now we will look at the structure of chlorophyll. Chlorophyll requires Mg2 plus to function, right? And chlorophyll A has CH3 in this R group, and chlorophyll B has CHO as this R group, and the porphyrin ring is shown in red. Okay, and this is the hydrophobic tail. Okay, now what about cytochrome C? Cytochrome C requires Fe2 plus for its function. Okay, cytochrome C, if you remember, this is also useful in this photosynthesis. So cytochrome C comes over here. Cytochrome BC complex is there. It is also made up of iron sulfur protein, and later on also in the electron transport chain cytochrome C is needed. Okay, now this is a very simple question. If you look at the structures, you will be able to identify exactly what molecule it is. Now this is a molecule that you have studied about very much. Okay, everyone knows this molecule. Everyone is aware of this reaction. Try to understand what is happening here and what enzyme and what cofactor may be involved. So if you know the enzyme, automatically you will understand what cofactor it uses. Okay, so try to answer the question. A is hexokinase and Mg2 plus, right? Very good. Yes, you guys are absolutely correct. See, this is the structure of glucose, and glucose is getting phosphorylated here, and it is forming glucose 6-phosphate. This is the 6th position. Phosphate group is getting added, and ATP is being utilized in this process because this is hexokinase enzyme, kinase after all, and Mg2 plus is required for its activity. Okay, and now we move on to another question. Now we actually studied about cupredoxin very recently, right? Cupredoxins are group of proteins which contain copper in their structure. Now one particular geometry is given and you have to tell me what this geometry is. This should be simple for you. Try to answer this. Okay. No, this is the answer to the previous question. Okay. Try to answer. What do you think is the geometry? Okay. Trigonal pyramidal. Right. Let's look at the answer. Yes, that is very correct. Cupredoxin has tetra-coordinated Cu. So Cu is coordinated by 4 different entities in its active site, and the ligands are 2 N-histidine. So this nitrogen from histidine is binding, and 1 S from the cysteine at its trigonal base, with 1 S from methionine at its long axis. It is a type 1 Cu center having trigonal pyramidal geometry. Okay, so if you are interested in this type 1 and type 2 copper center geometry, you can read about this more. Okay, right, we move on to the next question. Now this is a match the following question. So try to answer. Catalase. What does catalase do? What about peroxidase and rubridoxin and ferredoxin? So if you remember the figure of rubridoxin and ferredoxin, you will be able to answer the questions. Peroxidase you guys already know. Try to answer. Okay, you're saying it is D. Okay. Let's look at the answer. Yes, that is very correct. It is D. Okay, so peroxidase of course, this catalyzes oxidation of a wide variety of organic and inorganic substances using H2O. Catalase catalyzes the disproportionation of H2O2. So what is disproportionation? Disproportionation means it is a reaction in which the same molecule is simultaneously getting oxidized as well as reduced. So if you see here, it is getting oxidized and it is getting reduced. Okay. So this is what is catalyzed by catalase. Coming to rubridoxin, this is a single ion tetrathiolate protein, and unlike other FES protein, it does not have inorganic sulfur. Coming to ferredoxin, it has multiple iron centers bridged by inorganic sulfur. So if you go back to this diagram, you see that it is having multiple centers and it is bridged by inorganic sulfur. But then this does not have inorganic sulfur. Why? Because this sulfur is donated by the cysteine residue. Right? So this is definitely not inorganic. So in all the four cysteines, the sulfurs are getting donated. So therefore, your right answer would be D. Okay. Now we will talk about azurin. So azurin is a molecule which is found in bacteria, and you know about the geometry of azurin. Now you try to solve this question, which is false regarding azurin. It has two copper ions. It undergoes oxidation reduction between Cu1 plus and Cu2 plus. Each Cu is coordinated by two histidines and two cysteines. So you're saying that D is wrong. Achha. And oxidized form of azurin receives only one electron. Okay, let's see if D is the wrong answer. Now, the wrong one is actually 1 and 4. These are wrong. Okay, so these are false. Why is that so? Because if you look into the geometry of azurin, you will see that this is a tetrameric protein. And okay, so tetrameric protein, it has 4 different subunits and there are 4 Cu centers. Okay, so those Cu ions are coordinated by 2 histidines, these 2 histidines, and 3 cysteine residues at equatorial positions. And carbonyl oxygen of glycine and methionine. Right, sorry. So 2 histidine and the third one is a cysteine. All the 3 are in equatorial position, and then you have carbonyl and glycine and methionine over here. Right. So azurin is a protein containing copper ion that shuttles between +2 and +1 oxidation states, and cytochrome C is a protein in which the heme-bound iron shuttles between +3 and +2 oxidation state. So then 2 is right, it is not wrong, and it has only 1 copper ion, sorry, it has 4 copper ions, so this would be wrong, and your 4th one is also wrong. Okay, right? Now we will talk about, if you are having any doubts, you can read about this more. Okay, right. So we will talk about the cytoskeleton now. So that is all with your metalloproteins. Now we will talk about motor proteins. So cytoskeleton. This is actually the skeletal framework of the cell. So our skeleton gives us a definite shape. It keeps our structural integrity intact, right? And for the same, it is the same way with respect to a cell. The cytoskeleton is made up of 3 units. There are also more, For example, there is microtubule, there are microfilaments, and there are intermediate filaments which help in cell-to-cell connection. They are helping in cell division. They are helping in transport of substances across the cell and from one cell to another. So they are performing a wide range of functions. We will have a look at all of the components in the cell, in the cytoskeleton. Okay, so these are the 4-5 things that we will be focusing on are kinesin, myosin, dynein, and we will also be looking at actin, and we will be looking at some other components. So if you look at this, so kinesin here is the one whose image we saw in this figure. This is probably kinesin, and this is helping in carrying cargo across the microtubules. Okay, so it is very important that you remember the structures of these molecules, of these proteins. So this is dynein and this is myosin. Okay, now the cytoskeleton is a cellular component that helps to maintain the shape and internal organization of the cell. It also provides mechanical support and enables the cells to carry out essential functions such as division and movement. The cytoskeleton consists of 3 different types of protein fibers. They are microfilament, intermediate filaments, and microtubules. Their size distinguishes them, with microtubules being the thickest, we saw that in the figure, and the microfilaments being the thinnest. Okay, so what are microfilaments? Now, of the 3 types of cytoskeletal protein fibers, microfilaments are the narrowest, with a diameter of about 7 nanometer. Now, they are made up of several intertwined actin monomers. We will learn about actin in the next slide. Okay, resembling a double helix. As actin monomers are the principal component of the microfilaments, they are also known as actin filaments. Like actins, microfilaments also have directionality, that is a plus end and a minus end, structurally different plus and minus ends, right? So microfilaments provide mechanical support to the cell by forming a band just beneath the cell membrane. These filaments play very important roles. Okay. Now we will look at actins. So if you look at these actins, the monomeric form of actins, they are globular. They are round, small and globular. If you see this green structure, this is the structure where the myosin comes and attaches. Okay. So these G actins or globular actins, they come together, bind, and they form this double helical filamentous form of actin, which is a polymer. Okay. Then what happens? Then comes in other proteins. For example, tropomyosin. So tropomyosin is also a fibrous protein which comes and binds to this myosin binding region of the actin. And there are also a trimeric protein which is called troponin. Troponin is made up of 3 different types of proteins, 3 different components. We will learn about them in the next slide. So these also come and attach, okay, to the tropomyosin. So see, thin filament. The thin filament is made up of 3 proteins. One is your actin, then is your tropomyosin, and then there is the troponin. Okay, so actin, we just will now recall whatever we learned until now. So actin is a globular protein which has low molecular weight, and it occurs in 2 forms: monomeric G form, which is the globular actin, and polymeric F form, or the fibrous actin, and filamentous actin. So G actin polymerizes to form the F actin in the presence of magnesium ion, right? So tropomyosin is a fibrous molecule. The 2 filaments of tropomyosin run closely the entire length of the actin. At resting stage, it separates the actin and myosin by binding to the myosin binding site on the actin filament, and hence, and hence prevents the formation of cross bridges, which in turn prevents contraction of the muscle fiber. So we will see in the next upcoming slides how muscle contraction happens when myosin and actin come in contact with one another. Okay. To prevent constant muscular contraction, this tropomyosin is covering the myosin binding sites on the actin. Okay. Now we'll talk about troponin. So troponins are a complex protein and it masks the active binding sites for myosin on the actin filament. So troponin is a trimeric protein. It has 3 units which act as the binding site for 3 different components. The 3 units of troponin are firstly troponin I, which inhibits actin-myosin interaction and binds to other components of the troponins. Then we have troponin T. It is the binding site for tropomyosin. And then we have troponin C, which is the binding site for calcium. You know that calcium is very important for muscle contraction, right? Calcium is to be released from the sarcoplasmic uh, reticulum and then muscle contraction happens, right? Okay, right. Now, this is something which is a little detailed. Okay, so how this actin components, actin, the small g actins come together to make the long filamentous actin, that is using energy and using other proteins, that is given, it is called actin treadmilling. So assembly and disassembly constantly happens. So one end keeps on adding and the other end keeps on dissociating, and this is how it moves. Okay, so actin polymerization can be prevented using a lot of different chemicals. For example, phalloidin, and then you have cytochrome D. These are all inhibitor molecules. So for your GATE, for CSIR NET, for ICMR DBT, you will be asked questions on these inhibitors. So please study about these inhibitors, okay? So you will be able to study about the inhibitors from either Voigt and Voigt, from Ledinger's biochemistry. So it is given very well there. Okay, plus you can also refer to the websites that I have cited here and the different review papers, and then that will give you a very good understanding of this entire process. But read about the inhibitors, that is very important. Okay, so if you are interested, you can look at this, but right now we don't need this, so I am just skipping this part. And this, I hope you've understood that when calcium ions come, they bind to this troponin, and this is the myosin binding site that gets exposed. Then what happens? Then the myosin will come and bind, and then muscle contraction will happen. So you know about muscle contraction, I'm sure, and for today's class, I don't think there are many questions on muscle contraction, but then we will quickly go through it, okay? So based on the sliding filament theory, thick filaments of myosin move past the thin filaments of actin. So this is the thick filament and this is the thin filament, and they require actin-myosin binding site, ATP and calcium 2+. So tropomyosin and troponin work together to block the myosin binding sites normally when contraction is not happening. Then what happens? There is ATP involved, and calcium 2+ allows for myosin binding on the actin by shifting the troponin-tropomyosin complex. So calcium comes and binds to this troponin C, and then what happens? The troponin-tropomyosin complex shifts, exposing the myosin binding sites. Then what happens? Then the myosin head is ready to bind to the actin once it has hydrolyzed ATP. So this is what is happening. It has hydrolyzed ATP and it is now ADP plus PI, and then it is binding, right? So as soon as it is binding, this PI is released. Now a strong bond between actin and myosin is able to form. This is called a cross bridge, and a power stroke happens. ADP is released. Now the muscle is contracted and ATP binds myosin again, causing a release of the actin back to the relaxed state. So after stage 3 happens, the myosin head undergoes a large conformational change and it is actually binding to this actin, creating a power stroke, and ADP is released in the process and then it gets dissociated once again. Okay, so there is a constant cycle of this going on, and this is a reversible reaction. Okay, so now these are the different motor proteins found in nature: myosin, kinesin, dynein, flagellin, and ATP synthase. So you need to read about all the 5 because we have questions from all the 5 of these. Okay. What about myosin? Myosin is of course helping in cell muscle contraction. It is helping in cellular movement and cytokinesis. Read the mechanism of action as well. What about kinesin? This is a transport protein. Then we have dynein. This is also transport protein, but it is acting exactly opposite to that of kinesin. Okay. What about flagellin? Flagellin is found in flagella. It is helping in locomotion, helping in movement of the bacteria. What about ATP synthase? This is an enzyme which is helping in the synthesis of ATP from ADP and PI. Okay, read about these in details. Now we will look at dynein and kinesin. So if you remember the first image that we saw in our PPT, it was the image of kinesin possibly, and this is the image of dynein and kinesin. So what happens is they are both acting exactly opposite to one another. So kinesin moves from the cell body to the axon terminal and it carries cargo in this direction. This is called anterograde, that is in the forward direction. Now dynein, this moves along the microtubule and it carries cargo from the axon terminal to the cell body. So this is traveling in retrograde direction. Okay, you'll get asked questions on this, whether this is traveling in anterograde or retrograde direction. Right, so kinesins are moving along the microtubules, usually carry cargo such as organelles and vesicles from the center of the cell to its periphery. Okay, dyneins, they are important in sliding microtubules relative to one another during the beating of cilia and flagella on the surfaces of some eukaryotic cell. They carry the cargo from the periphery to the center of the cell. Okay, right. Now we will look at the structures of dynein and kinesin. If you are interested, you can go through this, but for now we don't need this as such. Okay, so we'll actually go through this. So see, kinesin, this is having a heavy chain and it is having a light chain. Okay, so and this is cytoplasmic dynein. So this is actually a multimer and this is how it travels. You can look at that later on if you're interested. Look at it in details. So we have the first question, which is which of the following is true? Myosins are actin-based motor system. Dyneins are actin-based motor system. Myosins are tubulin-based motor system, and kinesins are tubulin-based motor system. Which of the two sentences do you think are right? C. Okay, very good. C is right because we saw that myosin, this is attaching to actin and therefore it is a myosin. Myosins are actin-based motor system. Kinesins and dyneins, both of these are tubulin-based motor system. They are traveling, see, on the microtubule and performing their function. Right? Okay. Now we have another question, which is which of the following is true? Dyneins show retrograde transport. Kinesin show retrograde transport. Kinesin help to move the chromosome along the microtubule. The ATP bound state of kinesin weakly binds with the microtubule. Which of the following do you think would be right? Okay, you're saying that it is A. Let's look at the right answer. Yeah, that is true. Dyneins are retrograde, that is, it is moving from the moving towards the plus end, right? From plus to minus, and the ATP bound state of kinesin actually very strongly binds to microtubule, and therefore this would be wrong. This sentence is wrong. Now kinesin helps to move the chromosomes along the microtubule. That is true. Okay. Now this is something Very interesting. Just now we studied about this muscle contraction, right? So based on the diagram, try to understand this particular question and try to tell me which one do you think would be right. Okay, myosins are in a high energy state when their head is bent at an angle of 90 degrees. Myosins are in a high energy state when the head is bent at an angle of 45 degrees. Actins are in low energy state when the head is bent at an angle of 45 degree, and actins are in low energy state when the head is bent at an angle of 90 degrees. B. B is true. A is true. Okay, we have mixed answers. Okay, let's see. Myosins are in a high energy state when the head is bent at an angle of 90 degree. So myosins are in a low energy state when the head is bent at 45 degrees, and at high energy state when the head is bent at 90 degrees. So if we go back to the figure, if you look at this, you see that this is actually, this is 45 degree from this surface and it is becoming 90. This is actually the head. So head is bent at 90 degree with respect to this, right? This is a right angle. When is that happening? This is 45 degree. If you extend this line, this is going to be 45 degree. And when is this happening? See, so there is a power stroke. So power stroke means that very high energy state and the conformational change is happening. So when they are at high energy, they are 90 degree. They are at 90 degree. And normally they are at 45 degree when they are not at high energy. Okay. So then myosins are low energy when the head is bent at 45 degree and high energy state when the head is at 90 degrees. Okay. Oh, this is, it shouldn't be centigrade. It should just be degrees. Right. Now we move on to the next question. Next topic. So this is something very interesting. So you might have studied about the cilia and flagella. Right. In bacteria, there are these tiny extensions all over the surface of the bacteria. This is called cilia. So cilia actually goes through this kind of motion and helps the bacteria to travel. Okay. What about flagella? Flagella flips around and helps the bacteria to move. So both cilia and flagella are helping in locomotion. Cilia is also helping in different kinds of other things such as reproduction. It is also helping, let's say, in sticking to a surface and all that. So you can read about that. Now, this cilia and flagella, they are also motor proteins. They are made up of different subunits. So what happens is, there are these doublet microtubules that are arranged. There are 9 doublets present all over, and at the center there are 2. Okay, 2 central microtubules. So that is 9 plus 2 pattern. And these are having dyneins as arms. Plus there are radial spokes. So cytoplasmic dynein particles to the minus end of the microtubule. This is actually axonomal dynein, and this oscillates and is responsible for the beating of the cilia and flagella. Axonomal means dynein present in axon. Kinesin moves towards the plus end of the microtubule. You know that dynein is having retrograde transport. It is moving to the minus end, and kinesin is moving in the anterograde or towards the plus end of the microtubule. The myosin 2 binds to actin and produces motion by bending their neck region. So this is how the cilia and flagella move. Okay. Right. Now you can read about Nexin and everything else. Nexin is actually connecting these microtubules together. Okay. You can read about this in more details. For this particular question, I think this is all you need. So you try to answer this question. An image is given where you have X, Y and Z. X is something, Y is something, and this is the image of an axonal what? Kinesin or dynein. Okay, you're saying that it is. Acha, no, you haven't answered. Huh. So, okay, I'm getting D and A as answers. Okay, let's see. Okay. So D is the right answer. This is the image of an axonal dynein. X, Y, and Z are radial spokes. So this, if you remember from the figure, see, this green one is radial spoke. This round microtubules are there. Microtubule A and B together, doublet. And these tiny ones, they are actually dynein arms. So your Z should be dynein arms. Y should be microtubule doublet, right? And X should be radial spokes. And this is an axonal dynein given in this figure. Okay, right. Now we go to the last few questions. No, okay, we have a lot of questions left. Okay, anyways, so we'll continue. Now this is a question on hemoglobin and myoglobin. Now this is something that we have studied earlier. Just one statement is new here for you. So you must be able to answer this, right? So the rest of it is something that we have read in our previous classes. Try to see which is the false statement. Which statement is false regarding myoglobin and hemoglobin? Oxygen saturation, B is false. 2,3 ppg increases oxygen affinity of hemoglobin. Okay, so you're saying that it is false. Okay. Let's see. Achha, I'm getting two different answers. Iron binding sites are similar for both myoglobin and hemoglobin. Yeah, so B is the false answer. It decreases oxygen binding affinity. Okay, and iron binding sites are the same for both hemoglobin and myoglobin. If you come to this figure where we studied about the porphyrin ring, you will see that this and this is exactly the same. Right? The only difference is this functions by cooperativity. This you can look up what happens. Okay. Right. You can also read about the oxidation states. Okay. Right. Now, we will go through the enzymes involved in DNA replication at a glance. So, the first and the most important enzyme is DNA helicase. What does helicase do? You know that DNA is present as a helix. Now, what helicase does is, helicase unwinds the DNA and forms the replication fork. Now, what happens? The single-stranded binding proteins, these come and bind to the single-stranded open region of the replication fork and stabilizes them and prevents them from rewinding. Then we have topoisomerase. Topoisomerase is a very important enzyme because topoisomerase is a very surprising enzyme because in nature this actually makes cuts in the DNA. There are two types of topoisomerases. One is topoisomerase 1 which makes single-stranded cuts. Topoisomerase 2 which makes cuts and at both the strands of DNA. And what happens is it unwinds the DNA and it joins again. So it cuts and joins both, right? It has both the activities of cutting as well as joining. And that is why it is very, very special in nature. Okay, so this actually unwinds the DNA because as the replication fork happens, what happens is there is a very tight positive supercoil. So positive supercoil means very tight supercoil in the DNA that is happening because when you're opening this part is going to undergo a lot of stretch, right? And that is going to build up a lot of pressure, and because of, to prevent that, to release the pressure, this topoisomerase is releasing the supercoil in the DNA. There is DNA guidase. Achha, now there are two different things. One is positive supercoil and one is negative supercoil. Now what is that? Negative supercoil means it is reducing the supercoils. Okay, it is having less number of supercoil compared to its capacity. Positive supercoil means it is more supercoil and it is like very unfavorable. But negative supercoil is favorable by nature. So DNA guidase is an enzyme found in bacteria which introduces this negative supercoil in the DNA. Okay. Now, RNA primase. Primase means it is going to come and it initiates the new strand synthesis by adding the small single-stranded primers. You know about Okazaki fragments, right? So these primers get extended and they form the Okazaki fragments at one strand, and another strand there is a single primer, and then there is 5' to 3' synthesis, and you get a long DNA strand, right? So that is performed by DNA polymerase. So in bacteria it is DNA polymerase 3 that is doing this, fine. And these Okazaki fragments are again ligated by DNA ligase. That should be clear. Okay. Now we will try to answer one small question regarding this. Okay, which among the following is or are true or false about the DNA topoisomerases? It relieves strain caused by unwinding of the DNA by helicase and relaxes the coil. Topoisomerase 1 cuts both strands, topoisomerase 2 cuts one strand. Which of these do you think is false? Now you can expect many questions from this DNA topoisomerases and DNA guidases. Right. So you're saying that B is the right answer? Yeah, very good. That is right. So you can expect many questions from this portion. Okay. Topoisomerases, DNA guidases, linking number, all of these are very favorite questions of your examiners. Plus, they can ask you to calculate the twist, the read of a DNA, and then linking number of a DNA. So you must practice those. Okay. There are many numericals also, very simple, very easy numericals, very scoring. But then you need to understand what linking number is, when linking number can be changed, everything. I think we have this in our session also. I mean, maybe in the future sessions, we'll see. Okay. So see here, topoisomerase 1 cuts only one strand and topoisomerase 2 cuts both the strands. So type 1 topoisomerase, relaxation happens when the cut strand is reannealed. So it cuts and then joins. Similarly, type 2 topoisomerase breaks both the strands of DNA. You can see in this figure, and then reanneals, right? Okay. Now this is the same thing given but in greater details. You can go through this and you can see how this nick is joined and how the supercoiling is, the strain is basically released, and then there can be also catenated relaxed DNA, that is also possible. So you can go through this. Okay. Right. Now what is catenated DNA? Catenated means that this is there and this is there. This is how it is getting interlocked. That is called catenation. So when you break this and you insert this inside and you join it, that is how catenation happens. You read about that if you're interested. Right. So now there is one more match the following question. This is all about DNA polymerase, helicase, guidase and ribosome. See what is happening. Polymerase. Polymerase. What does polymerase do? What does helicase do? What does guidase do? And what does ribosome do? Yes, very good. Because the last one directly matches with A. Right? And there is only one answer for this. So you don't really need to read the other options. So B is the right option. Polymerase, DNA polymerase and RNA polymerase, DNA polymerase catalyzes the addition of deoxynucleotides, and RNA polymerase uses single-stranded DNA template to synthesize a complementary strand of RNA. What about helicase? This actually derives energy from ATP to move along the nucleic acid and separate the two annual nucleic acid strands. DNA kinase catalyzes the DNA in supercoiling or relaxation and introduces negative supercoils into the DNA at the expense of ATP hydrolysis. Ribosome, of course, you know that it is involved in translation of proteins. Right. Now we have almost come to the end of the class. There are a few more questions and then we'll watch a very nice and interesting video. Okay. This is the video of motor proteins walking. So I've saved this video for you and I really wanted to show this to you. We'll see this at the end of the class. So, now this is one more question. See, there are 3 plots given for partial pressure of oxygen. The plots of saturation fraction theta and partial pressure of oxygen of hemoglobin at different pH 1, 2 and 3 are given below. The order of 1, 2 and 3 will be A. Okay, great. So at lower pH, oxygen affinity for hemoglobin decreases, and therefore, if that is the trend, then we can say that 3 is the lowest pH in the plot, right? And it shows lowest oxygen saturation at a particular pH. Okay. Now we have one more question, which is, this is an iron sulfur protein along with molybdenum. And this is a normal iron sulfur protein. Okay, so what exactly is happening here that they have asked you? Electron transfer takes place from cluster 1 to 2 and N2 binds with cluster 1. Sorry, N2, this one from histidine. Then electron transport takes place from cluster 2 to 1 and then N2 binds with cluster 1. So what do you think is happening here? D. Okay. Then the right answer is B. Electron transfer takes place from FeMO cluster, which is this one, cluster 2, sorry. FeMO should, I think, be cluster 1, 2M cluster 1 of nitrogenase. N2 binds to cluster 1 and then gets reduced to NH3. Okay, we'll have a look at this. You can confirm this with the NPTEL team if there is any doubt. Okay. Now this is all about the nitrogenase enzyme complex. This is the Fe protein and this is FeMO protein. And you can see that pyridoxine is undergoing from the reduced state, it is getting converted into the oxidized state. Similarly, here also redox reactions are happening and redox reactions are happening here, and then we see that the products are getting released. So from this cluster to this cluster, electron transport is definitely happening. So from this Fe cluster, which is cluster 2, to this cluster, electron transport is happening, and then what is happening? You're getting ammonia as the product. Okay, right. Now we come to this video. You tell me if you are able to see it. So here we will see the kinesin walking along the microtubule. So see, this is how it walks. Can you see the video? This is how it is walking. It is exactly walking like a human, and it is carrying a cargo. And this is walking on a microtubule, which is constantly getting formed and it is getting degraded. So that is very interesting, I think. You can read about this more if you're interested. Okay. Thank you so much. Here are the different materials the references that are there. You can go through them. We have 2 pages of references today. Thank you so much for being with me all through. Okay, if you have any questions, you can ask me. Tomorrow also we have a class in which we will be discussing week 9 question answers. Okay. Thank you so much for your time. If there are no more questions, we can end the session today. Thank you so much. Some of the questions are repeated from the previous week 8. Yeah, this is actually the week 8 video. We hadn't discussed week 8 question answers, no, last week. That is why the class had been postponed, and today we discussed all the questions from week 8. Tomorrow we will be discussing the questions from week 9. So this is supposed to have questions from week 8. Okay. Week 9 will be necessary. Okay, great. We'll discuss week 9 in details then. All right. Thank you so much for your time. We'll be ending the session today. I'm ending the session for everyone. Good night to you too.